when you hear lipids, does your mind automatically jump to fats? Because if it does, you're only seeing really a tiny fraction of the picture. We're talking about molecules that are absolutely everywhere in our bodies, doing a ton of different jobs. They're not just stored energy. They're literally building our cells, shuttling messages around.
Yeah.
They're kind of a big deal.
Exactly. And what's fascinating is just how varied lipid biochemistry is. We're going to start with some relatively simple building blocks, fatty acids, and work our way up to these incredibly complex structures, steroids. We'll be exploring the key roles they play in keeping us alive and kicking. The goal isn't just to scratch the surface. It's really to give you the same kind of solid understanding experts in the field rely on.
So buckle up for a thorough unpacking of the world of lipids. We're going to look at how they're categorized, what their many different jobs are, and how they impact our health. We've got a solid stack of research to guide us on this journey.
Yeah, and the first thing to understand is that the word lipid covers a huge range of molecules. They can be simple, complex, or derived. Let's start with the simple ones. These are basically what you get when fatty acids hook up with different types of alcohols. Think of it like a basic Lego set almost.
Okay, so within the simple lipids bucket, we've got fats and oils. What's the real difference? I mean, besides one being solid and the other liquid?
Good question. Well, both fats and oils are made of fatty acids linked to glycerol. It's the type of fatty acids they contain that determines whether they're solid or liquid at room temperature. Fats tend to have more saturated fatty acids, which pack together tightly, making them solid like butter.
Right.
Oils have more unsaturated fatty acids, and those kinks, the double bonds, prevent tight packing. That makes them liquid, like olive oil.
Right. The unsaturated ones have those little bends that keep them from stacking neatly. Got it.
Right.
And then there are waxes. Always forget about waxes.
Waxes. They're also simple lipids, but instead of glycerol, they use larger, higher molecular weight monohydric alcohols. This makes them very water resistant and gives them that characteristic hard protective texture. Think of the coating on plant leaves or beeswax.
Okay, that makes sense. Right, complex lipids now. Sounds like we're leveling up in lipid complexity here.
Absolutely, we are. Complex lipids are also fatty acids linked to an alcohol, but they have extra chemical groups tacked on. These extra bits give them different properties and jobs. The main categories here are phospholipids, glycolipids, and a few other interesting types.
Phospholipids first. I know these are crucial for cell membranes. It's like what makes them the perfect barrier to keep the cells safe.
Exactly. You nailed it. Phospholipids all contain a phosphoric acid residue, which often has a nitrogen-containing base attached, like choline, for example. Now, the alcohol backbone can be either glycerol, making them glycerophospholipids,
Okay.
or it can be sphingosine, which contains an amino group, creating sphingospholipids.
Glycerophospholipids. The name phosphatidic acid rings a bell. Like it's a key player somewhere.
It is. Phosphatidic acid is a key starting point, yeah. An important intermediate in making other glycerophospholipids. You can think of it as the foundation upon which many other phospholipids are built. It's present, but usually in quite small amounts itself.
And then the sphingospholipids use sphingosine. Sphingomyelin is the one I remember.
Sphingomyelin, yeah, that's a prime example. It's found in cell membranes and makes up a big part of those specialized areas called lipid rafts. But it's also super important in the myelin sheath, which insulates nerve fibers and helps them transmit signals quickly. Very critical there.
Okay, that's phospholipids. Now, glycolipids. The glyco part makes me think of sugar.
You got it. Glycolipids, or glycosphingolipids, are made of a fatty acid, sphingosine, and one or more sugar molecules. These are abundant in nerve tissues and stick out on the outer surface of cell membranes. The sugar part contributes to the glycocalyx, kind of like a sugar coating that helps cells recognize each other.
So cell-to-cell communication. Are there other complex lipids that don't fit neatly into those categories?
Yes, there are a few others. Sulfolipids contain sulfate groups, and aminolipids contain amino groups. Lipoproteins, which we'll definitely talk about more later when we discuss how lipids travel in the blood, are also technically complex lipids because they're lipids bound to proteins.
All right, simple and complex down. Now derived lipids. Sounds like the leftovers after you break down the others.
In a way, yes. Derived lipids are what you get when you hydrolyze or, break apart simple and complex lipids using water. This category includes things like free fatty acids, glycerol, steroids like cholesterol, other alcohols, ketone bodies used for energy, hydrocarbons, lipid-soluble vitamins like A, D, E, and K, and even some hormones.
It's interesting that some of these leftovers can also be the starting materials. A bit circular.
Exactly. Free fatty acids and glycerol released when triacylglycerols are broken down can then be used to build new lipids. It's a constant cycle of building up and breaking down. Very dynamic.
You mentioned neutral lipids earlier. What makes a lipid neutral? Just no charge.
That's basically it. Neutral lipids don't have an electrical charge at physiological pH. The main examples are acylglycerols like mono, digly, and triacylglycerols, and also cholesterol and cholesterol esters, which are cholesterol with a fatty acid attached.
Okay, let's zoom in on fatty acids. They're described as aliphatic carboxylic acids. That sounds pretty textbooky.
It does sound a bit formal, yeah. Let's unpack that. Aliphatic just means the carbon chains are straight or branched, not in a ring structure like benzene. Carboxylic acid means they have that COH group at one end, which is what makes them acidic. They're usually found esterified, meaning linked up, as part of fats and oils, but they also circulate in the blood as free fatty acids, which is how they're transported around the body.
I've heard that natural fatty acids usually have an even number of carbon atoms. Why is that? Is there a reason?
That's generally true, yeah, and it's because of how they're synthesized in the body. They're typically built by adding 2 carbon units, specifically from acetyl-CoA, one after another. Okay. And then we have saturated versus unsaturated. It's all about those double bonds, right? Or lack thereof.
You got it. Saturated fatty acids are saturated with hydrogen because they have no double bonds between the carbon atoms in their chain. Think of them as straight chains. Unsaturated fatty acids have one or more double bonds, and those double bonds create kinks or bends that affect how the fatty acids behave and pack together.
Naming these things seems like a whole language of its own. All the oics and inoics.
It can seem that way. The systematic name is based on a hydrocarbon with the same number of carbons. For saturated fatty acids, we change the ending from E to anoic, so octane, an 8-carbon chain, becomes octanoic acid. For unsaturated ones, we use enoic, so octadecane, 18 carbons, becomes octadecanoic acid, which you probably know better as oleic acid.
And carbon numbering starts at the carboxyl end, the NACN COAH, always. Exactly. That carboxyl carbon is always carbon number 1. The next carbons can be labeled with Greek letters. Alpha is carbon 2, beta is 3, gamma is 4, and so on. The very last carbon in the chain, the methyl group at the end, is called the omega carbon, sometimes written as ag, or just the N carbon.
And there are different ways to show where the double bonds are located. Delta and omega notations. How does that work?
Yes. The delta notation counts from the carboxyl end, carbon 1. So at F9 means there is double bond starting at carbon 9 between carbons 9 and 10. Omega notation, or N, counts from the other end, the omega or methyl end. So a 9 or N9 means the first double bond, counting from the omega end, starts 9 carbons in. For example, oleic acid has 18 carbons and 1 double bond. It's 18.1. That double bond is between C9 and C10. So you can write it as 18.1 N9, or counting from the omega end, it's also an omega-9 fatty acid, so 18.1 N9 or 18.1 N9.
Okay. The 18.1 tells you 18 carbons, 1 double bond. Makes sense.
Exactly. Number of carbons, colon, number of double bonds.
So that omega number is how we get the omega-3, omega-6, and omega-9 fatty acid families that we always hear about in nutrition.
Precisely. Animals, including us, have limitations. We can't introduce a new double bond just anywhere. We can generally only add new double bonds between an existing double bond and the carboxyl end. This means if you start with an omega-3, omega-6, or omega-9 fatty acid, you stay within that family as you modify it. That's why these families are distinct and why some, like omega-3s and omega-6s, are essential in the diet.
Okay, let's look at saturated fatty acids. You said they're built from acetic acid, just adding Danix CH2 units.
That's a good way to think about the series, yeah. Acetic acid has 2 carbons, add a CH2 and you get propionic, 3 carbons, add another, you get butyric acid, 4 carbons found in butter. Keep going, you get longer chains. Caproic 6, lauric 12, and coconut oil, mespristic 14, palmitic 16, and stearic 18. Palmitic and stearic are really common in animal and plant fats. Most are straight chains, but you do find some branched chain fatty acids in nature too.
Now, unsaturated fatty acids.
Yep.
We've got mono and polyunsaturated. Pretty straightforward naming, right? 1 double bond versus 2 or more.
Exactly. Monounsaturated fatty acids, or mononoic acids, have just 1 double bond. Polyunsaturated, or polynoic, have 2 or more. And then there's a really important subgroup derived from 20-carbon polyunsaturated fatty acids, the eicosanoids.
Eicosanoids. Prostaglandins, leukotrienes. These are like local signaling molecules, I remember that.
Absolutely. Prostaglandins, prostacyclins, thromboxanes, collectively called prostanoids, plus leukotrienes, and lipoxins are all eicosanoids. Prostaglandins are found in almost every tissue and act like local hormones, produced right where they're needed. They can affect everything from inflammation and blood pressure to smooth muscle contraction and blood clotting.
Thromboxanes are slightly different structurally.
Yeah, thromboxanes have a slightly different ring structure within the molecule compared to prostaglandins. They're particularly important in platelet aggregation, blood clotting. And it's interesting that different 20-carbon precursor fatty acids, like arachidonic acid versus EPA, can make different series of eicosanoids, which can then have slightly different, sometimes opposing, biological effects.
And leukotrienes and lipoxygens are made in a different way.
Right.
Not the same pathway.
Right. They're made via the lipoxygenase pathway, not the cyclooxygenase pathway that makes the prostanoids. Leukotrienes are potent mediators of allergic reactions and inflammation, causing bronchoconstriction, which is why they're heavily involved in asthma. Lipoxygens tend to have anti-inflammatory roles.
Okay, here's a key point you mentioned. Most naturally occurring unsaturated fatty acids have cis double bonds. What's the significance of that? Why cis?
It's all about the shape. Saturated fatty acids with no double bonds tend to be relatively straight, forming a sort of zigzag pattern. They can pack together quite neatly, but introducing a double bond restricts rotation. It can be either cis or trans. A cis double bond creates a definite bend or kink in the fatty acid chain.
So cis and trans refer to how the rest of the carbon chain is oriented around that rigid double bond.
Exactly. In a cis configuration, the acyl chains, the carbon chains on either side, are on the same side of the double bond. That creates that kink, about a 30-degree bend. In a trans configuration, the chains are on opposite sides, so the molecule ends up being much straighter, more similar in shape to a saturated fatty acid. Oleic acid is the classic cis example, while oleic acid is its trans isomer.
So almost all the unsaturated fatty acids made in our bodies and found in natural unprocessed foods are cis, meaning they're bent.
Yes, overwhelmingly cis. This bending has huge implications for how these fatty acids pack together, especially in cell membranes. For example, arachidonic acid, which has 4 cis double bonds, is actually bent into a sort of U shape. This prevents tight packing and is crucial for maintaining the fluidity of membranes.
But we do encounter trans fats sometimes, right? They're not all cis.
That's true. They occur naturally in small amounts in the fat of ruminant animals, like cows and sheep. This is due to microbial action in their stomachs. But the major source of trans fats in many diets, historically at least, came from partially hydrogenated vegetable oils. This industrial process, used to make liquid oils more solid and stable, like from margarine or shortening, unfortunately converts some of the natural cis bonds into trans bonds.
And we know now that these industrial trans fats are pretty bad for us.
Yes, definitely. Extensive research has linked dietary intake of trans fatty acids to an increased risk of cardiovascular disease, raising bad LDL cholesterol and lowering good HDL cholesterol. There are also links to inflammation, diabetes, and potentially some cancers. Because of this, many countries have implemented regulations to limit or ban industrial trans fats in foods. The World Health Organization has even called for their complete elimination from the global food supply.
So the physical properties of fatty acids, like melting point, really depend on both the chain length and the degree of unsaturation, those double bonds.
Absolutely. It's a clear relationship. Melting points increase with longer chain length, but decrease significantly with unsaturation, especially with cis double bonds.
So that explains why triacylglycerols, or triglycerides, the storage form with 3 fatty acids attached to glycerol, can be solid fats or liquid oils. It depends on the fatty acids they contain.
Exactly. Triacylglycerols containing mostly long-chain saturated fatty acids, like in beef fat, are solid or semi-solid at room temperature because those straight chains pack well. Triacylglycerols rich in polyunsaturated fatty acids, like in sunflower oil, are liquid because those kinks prevent tight packing. And it's not random. Natural isoglycerols have specific mixtures of fatty acids tailored for their functions. For instance, lipids in membranes need to maintain fluidity, so they have a higher proportion of unsaturated fatty acids. Animals living in cold climates also tend to have more unsaturated fats to keep their body fats from solidifying.
We hear a lot about omega-3 fatty acids being good for you, particularly for inflammation. What's the story there?
Yes, there's a lot of focus on long-chain omega-3s, like alpha-linolenic acid, ALA, found in flax seeds, and especially eicosapentaenoic acid, EPA, and docosahexaenoic acid, DHA, found mainly in fatty fish. They seem to have significant anti-inflammatory effects. One proposed mechanism is that they compete with omega-6 fatty acids, like arachidonic acid, for the enzymes that produce eicosanoids. The eicosanoids produced from omega-3s tend to be less inflammatory or even anti-inflammatory compared to those derived from omega-6s.
And that's why they're being investigated for treating various chronic diseases.
Precisely, because chronic inflammation is thought to be a key underlying factor in many major diseases like cardiovascular disease, rheumatoid arthritis, Alzheimer's disease, and some cancers. There's intense investigation into the therapeutic potential of omega-3s. Current evidence suggests they can be beneficial, particularly for cardiovascular health, although the exact extent and mechanisms are still being actively researched.
Okay, let's talk a bit more about triacylglycerols, or triglycerides as they're often called. We've established that those are the main way we store fatty acids, our main energy reserve.
That's right. They are esters of the alcohol glycerol with 3 fatty acids. Simple structure, highly efficient energy storage because they're very hydrophobic and packed densely. We also find monoacylglycerols, 1 fatty acid, and diacylglycerols, 2 fatty acids, in tissues. These are important intermediates in the synthesis and breakdown pathways of triacylglycerols, and diacylglycerols also function in signaling.
And you mentioned earlier there's a special way of numbering the carbons on glycerol, the SN system. Why is that needed?
Yes, the SN stands for stereochemical numbering. It's needed because the glycerol molecule itself is prochiral when the fatty acids at positions 1 and 3 are different, meaning carbons 1 and 3 aren't chemically identical in that context. So to be precise about which carbon we're talking about, especially when enzymes act on them, we use the SN numbering. By convention, the L-glycerol 3-phosphate configuration is used as the reference. Enzymes that act on glycerol lipids are often very specific, for example, acting only on the SN1 or SN3 position, like glycerol kinase, which specifically phosphorylates SN3.
Got it. Back to phospholipids, key part of membranes, you said, the main structural lipids.
Absolutely. They are the fundamental building blocks of all biological membranes, forming that lipid bilayer structure. The most common type are the glycerophospholipids, which are derivatives of phosphatidic acid. Remember, that's glycerol esterified to 2 fatty acids and a phosphate group. Phosphatidic acid itself is an important intermediate in synthesizing other phospholipids and triacylglycerols, but it's usually present in fairly low concentrations in tissues.
And then there are the sphingolipids, like sphingomyelin, which also play a structural role in membranes, especially in certain types like nerve cells.
Yes. In sphingolipids, the backbone isn't glycerol, it's sphingosine, which is a more complex amino alcohol. Sphingomyelin has a phosphate group attached to sphingosine, usually with choline as the head group. Both glycerophospholipids and sphingolipids generally fit the pattern needed for membranes. They have 2 long hydrophobic tails that bury themselves in the core of the bilayer and a polar hydrophilic head group that faces the aqueous environment.
How do the tails compare? Are they always fatty acids?
Good question. In glycerophospholipids, both tails are fatty acids, esterified to the glycerol backbone at SN1 and SN2. In sphingolipids, like sphingomyelin or glycolipids, one tail is a fatty acid attached to the amino group of sphingosine via an amide bond forming ceramide. The other hydrophobic tail is actually part of the long hydrocarbon chain of the sphingosine molecule itself.
Let's talk about some specific glycerophospholipids. Phosphatidylcholines, often called lecithins, are pretty common, right?
Yes, they're typically the most abundant phospholipids in the cell membranes of animals and plants. They're also a major storage form of choline in the body. Choline is important for various things, including making the neurotransmitter acetylcholine, and it can also be a source of methyl groups for metabolic reactions. A specific type, dipalmitoyl lecithin, is a major component of lung surfactant, that substance that reduces surface tension in the alveoli and keeps our lungs from collapsing when we breathe out. Its deficiency in premature infants causes respiratory distress syndrome. Structurally, phosphatidylcholines often have a saturated fatty acid at the SN1 position and an unsaturated one at SN2, contributing to membrane fluidity.
What about phosphatidylethanolamine and phosphatidylserine, similar but different head groups?
Exactly. They're also very important glycerophospholipids found in cell membranes. They have the same basic structure as phosphatidylcholine, but with ethanolamine, or the amino acid serine, attached to the phosphate group instead of choline. Phosphatidylserine is particularly interesting because it's normally kept on the inner leaflet of the plasma membrane, but if it flips to the outer leaflet, it acts as a signal for programmed cell death, apoptosis basically telling scavenger cells, eat me.
Wow, okay. And phosphatidylinositol, involved in signaling.
Yes, very much so. Phosphatidylenostatol contains a specific 6-carbon sugar alcohol isomer called myoanoacetyl as its head group. While it's a relatively minor component of membranes overall, its phosphorylated forms, especially phosphatidylenoacetyl 4-milli-5-bisphosphate or PIP2, are crucial precursors for intracellular signaling molecules. When certain cell surface receptors are activated, PIP2 is cleaved by an enzyme called phospholipase C into two second messengers, diacylglycerol, DAG, and inositol trisphosphate, IP3, which trigger downstream cellular responses.
Cardiolipin is another one specifically associated with mitochondria, I think.
That's right. Cardiolipin is a unique double phospholipid. It basically has two phosphatidic acid backbones linked by a glycerol molecule. It's found almost exclusively in the inner mitochondrial membrane. It's absolutely essential for the proper structure and function of mitochondria, particularly for the complexes involved in electron transport and ATP synthesis. Decreased levels or alterations in cardiolipin structure have been linked to mitochondrial dysfunction in aging and various diseases like heart failure, certain metabolic disorders like Barth syndrome, and even cancer.
What about lysophospholipids like lysophosphatidylcholine? They're missing a fatty acid.
Exactly. These are glycerophospholipids that have had one of their two acyl fatty acid tails removed, usually by an enzyme called phospholipase A2. So lysophosphatidylcholine or lysolecithin is only one fatty acid chain. They are important intermediates in the metabolism and remodeling of phospholipids. They also have detergent-like properties and can be involved in signaling. Lysolecithin is also found in oxidized lipoproteins and has been implicated in the development of atherosclerosis.
And finally, plasmalogens. These sound a bit different. Etherlink.
Yes, plasmalogens are a significant subclass of glycerophospholipids, making up maybe 10–30% of the phospholipids in brain and heart muscle tissue. Their defining feature is an ether linkage, OCH-CH, at the SN1 position of the glycerol backbone, instead of the usual ester linkage, OCO. The group attached via the ether link is typically an unsaturated alcohol. The head group is often ethanolamine, but choline, serine, or inositol can also be found. Their exact roles are still being fully elucidated, but they're thought to be important for membrane structure, perhaps involved in forming lipid rafts, protecting against oxidative stress, acting as endogenous antioxidants, and maybe in cell signaling.
Okay, so we've covered the main glycerophospholipids and mentioned sphingomyelin. Just to recap the core difference in structure.
Sure. Both main classes, glycerophospholipids and sphingolipids like sphingomyelin, have that amphipathic structure essential for membranes. Two hydrophobic tails and a polar head group. But in glycerophospholipids, the backbone is glycerol, and both tails are fatty acids attached by ester links, usually. In sphingolipids, the backbone is sphingosine. One tail is a fatty acid attached by an Mi bond, and the other tail is part of the sphingosine molecule itself.
Now, glycolipids. You mentioned earlier their importance in nerve tissues and on the cell surface, the sugar-containing lipids.
Exactly. Glycolipids are lipids with one or more carbohydrate chains attached. The major type in animal tissues are the glycosylphingolipids. These all contain ceramide that's sphingosine linked to a fatty acid plus one or more sugar residues. They are widely distributed, but particularly abundant in nervous tissue, like the brain. They are primarily found on the outer leaflet of the plasma membrane, and their carbohydrate portions extend outwards, contributing significantly to the cell surface carbohydrates, or glycocalyx, which is involved in cell recognition and interactions.
We talked about galactosylceramide earlier as an example.
Yes, galactosylceramide is one of the simplest and most common glycosphingolipids. It's a major lipid in brain and other nervous tissue, less so elsewhere. It characteristically contains C24 fatty acids, like cerebronic acid. It can be further modified by adding a sulfate group to the galactose sugar, forming sulfogalactosylceramide, often just called sulfatide. Sulfatides are found in high concentrations in myelin.
And glucosylceramide. Similar, but with glucose.
Right. Glucosylceramide has glucose instead of galactose linked to the ceramide. It's actually the predominant simple glycosphingolipid in extraneural tissues outside the nervous system, though it's also found in the brain in smaller amounts. Importantly, glucosylceramide serves as the precursor for most of the more complex glycosphingolipids, including the gangliosides.
Gangliosides sound complex just by the name.
They are indeed the most complex glycosphingolipids. They are derived from glucosylceramide by adding more sugar units, and crucially, they contain one or more molecules of sialic acid. The principal sialic acid in humans is N-acetylnerminic acid, or NUAC. Gangliosides are found in highest concentrations in the nervous system, particularly in the gray matter of the brain. They function in cell recognition, adhesion, communication, and also act as specific receptors for certain hormones and bacterial toxins, like the cholera toxin.
There's a shorthand nomenclature for gangliosides, like GM3 and GM1. What does that mean?
Yes, it helps navigate the complexity. G stands for ganglioside. The next letter, like M, D, T, or Q, indicates how many sialic acid molecules are present. Mono, D, tri, or quatra, 4. The number, like 1, 2, 3, relates to the migration pattern on thin layer chromatography, basically indicating the relative size and complexity of the carbohydrate chain. GM3 is the simplest ganglioside, consisting of ceramide, glucose, galactose, and one NUAC molecule. GM1 is derived from GM3 by adding more sugars, and it's particularly famous because it acts as the receptor for cholera toxin in the human intestine. Binding of the toxin to GM1 triggers the events leading to cholera symptoms.
Okay, let's switch gears and move on to steroids. Cholesterol is obviously the most famous one, often with a bit of a bad reputation in popular culture.
Yes, it gets a lot of bad press due to its association with heart disease when levels are high in the blood, but it's absolutely essential. We literally cannot live without it. Cholesterol itself is a crucial structural component of amyl cell membranes, helping to regulate fluidity. But perhaps even more importantly, it serves as the precursor for synthesizing a whole host of other vital molecules.
Like what? What comes from cholesterol?
Well, bile acids, which are critical for digesting and absorbing fats in the intestine, all the adrenocortical hormones like cortisol, stress hormone, and aldosterone regulate salt balance, all the sex hormones, estrogens, progesterone, androgens like testosterone, also vitamin D, which we synthesize from a cholesterol derivative in our skin using sunlight, and even cardiac glycosides, which are plant-derived but structurally related compounds that affect heart muscle contraction.
Wow, that's a diverse list. Do all these steroids share a common basic structure?
Yes, they do. All steroids are based on a characteristic 4-ring structure, often called the steroid nucleus. It consists of 3 6-membered rings, labeled A, B, and C, fused together with one 5-membered ring, D. This core structure is called cyclopentanoperhydrophenanthrene, if you want the full chemical name. Phenanthrene is the 3 6-membered rings, and cyclopentane is the 5-membered ring.
And the carbons are numbered in a specific way.
Yes, there's a standard numbering system for the 17 carbons that make up the 4 rings, plus any carbons and side chains attached to the rings.
I notice in diagrams, sometimes the rings look like simple hexagons, sometimes they have double bonds drawn in.
That's an important distinction. When you see simple hexagons, it usually represents saturated rings, where all the carbons have single bonds and are saturated with hydrogens. If there are double bonds within the rings, like in the aromatic benzene ring, they are explicitly drawn. Most steroids also have methyl groups, CH3, attached at positions 10 and 13. These are carbons 19 and 18, respectively, and often a side chain of varying length attached at position 17 on the D ring.
What's the difference between a steroid and a sterol?
Good question. Sterols are a subgroup of steroids. Specifically, they are steroids that have a hydroxyl OH group, usually at position 3, and lack any carbonyl O or carboxyl dexo-OH groups. Sterol names typically end in ol, like cholesterol or ergosterol.
Steroids have complex 3D shapes. Stereoisomerism must be important here.
Absolutely. Because of the multiple asymmetric carbon centers and the way the rings are fused, steroids exhibit extensive stereoisomerism. The saturated rings are not flat. They adopt non-planar conformations, primarily the chair form, which is more stable than the boat form. Almost all natural steroids have their rings in the chair conformation.
And the way the rings are joined together matters too, cis and trans junctions.
Yes, the fusion between adjacent rings can be either cis or trans. This depends on the relative orientation of the bonds connecting the rings. In most natural steroids, the B, C, and CD ring junctions are trans, which results in a relatively flat, rigid structure overall. The AB junction, however, can be either cis or trans, leading to different overall shapes.
And then substituents sticking off the rings have orientations too, alpha and beta.
Exactly. Substituent groups attached to the ring system can point either below or above the general plane of the rings. By convention, bonds pointing below the plane are designated as ASA and are drawn with broken or dashed lines. Bonds pointing above the plane are designated as eta and are drawn with solid, often thickened lines. For instance, the crucial hydrogen atom at position 5 determines the AB ring junction. If it's AS pointing down AB trans, you have a 5-tetroid. If it's ASIS pointing up AB cis, you have a 5-steroid. The angular methyl groups at C10 and C13 are pretty much invariably AT.
Okay, so cholesterol itself.
Yeah.
What are its key features?
Cholesterol has the basic steroid nucleus, a hydroxyl group at C3, making it a sterol, A double bond between C5 and C6 in the B ring, those two methyl groups at C10 and C13, and an 8-carbon branched side chain at C17. As we said, it's widely distributed in all body cells, but especially abundant in nervous tissue. It's a major constituent of plasma membranes, influencing their fluidity, and also a key component of plasma lipoproteins, which transport lipids in the blood. Inside cells and in lipoproteins, it's often found in an esterified form, where the hydroxyl group at C3 is linked to a long-chain fatty acid forming a cholesterol ester. This is even more hydrophobic than free cholesterol. And importantly, cholesterol is synthesized by animals. It's generally not found in plants or bacteria.
What about ergosterol? That's found in plants, right?
Ergosterol is found in plants and yeast, yes. It's structurally similar to cholesterol but has some differences, notably an extra methyl group and more double bonds, including one in the B ring and one in the side chain. It's significant because it's the dietary precursor to vitamin D2, ergocalciferol. When ergosterol is exposed to ultraviolet UV light, the B ring opens up, converting it into vitamin D2. This is similar to how 7-dehydrocholesterol in our skin is converted to vitamin D3 cholecalciferol by sunlight.
And polyprenoids, you mentioned they're related but not actually steroids.
Correct. Polyprenoids are another large class of lipids, but they don't have the steroid ring structure. However, they share a common biosynthetic origin with cholesterol. They're both synthesized from 5-carbon isoprene units. Cholesterol synthesis involves assembling these units and then cyclizing them to form the steroid nucleus. Polyprenoids are typically long chains formed by linking multiple isoprene units together. Examples include ubiquinone, coenzyme Q, which is crucial in the mitochondrial respiratory chain for electron transport. Dolichols are very long-chain polyprenols involved in adding sugar chains to proteins during glycoprotein synthesis. And many important plant-derived compounds are polyprenoids or derived from them, like rubber, camphor, the fat-soluble vitamins A, E, and K, vitamin D is steroid-derived, and carotenoids like carotene, provitamin A.
Okay, let's move on to something that sounds potentially damaging. Lipid peroxidation. What is that?
It is potentially damaging, yes. Lipid peroxidation is essentially the oxidative degradation of lipids, particularly polyunsaturated fatty acids, PUFAs. It's a process where lipids react non-enzymatically with oxygen, leading to the formation of lipid peroxides and hydroperoxides. This process is responsible for the spoilage of foods containing fats and oils, causing rancidity. But more importantly, it also occurs in our bodies and can cause significant damage to cell membranes and other structures. It's implicated in the pathology of numerous diseases, including cancer, inflammatory conditions, atherosclerosis, and it's thought to contribute to the aging process.
What drives the damage? Free radicals.
Exactly. The damage is primarily caused by free radicals. These are highly reactive chemical species that have one or more unpaired electrons in their outer shell. They readily attack other molecules like DNA, proteins, and especially the PUFAs, our cell membranes, stealing electrons and initiating chain reactions. Reactive oxygen species, or ROS, are a major type of free radical involved. These include peroxyl radicals, OR, alkoxyl radicals, or R, and the highly reactive hydroxyl radical, OH. These are produced during the formation of peroxides from PUFAs.
Why are PUFAs particularly susceptible?
It's because of the methylene CH2 groups located between the double bonds in PUFAs. The hydrogens on these carbons are relatively easy to abstract or remove, initiating the peroxidation process.
You said it's a chain reaction. How does that work?
Yes, it typically proceeds as a free radical chain reaction involving 3 main phases. 1, initiation. An initial free radical, let's call it X, perhaps from environmental exposure or normal metabolism, attacks a PUFA, RH, abstracting a hydrogen atom and forming a fatty acid radical. 2, propagation. This fatty acid radical is unstable and reacts very quickly with molecular oxygen, O2, to form a lipid peroxyl radical, ROO. This peroxyl radical is itself reactive and can attack another PUFA molecule, ROH, abstracting a hydrogen to form a lipid hydroperoxide, ROH, and a new fatty acid radical. This new radical can then react with oxygen, continuing the chain. So one initiating event can lead to the damage of many PUFA molecules. 3, termination. The chain reaction eventually stops when 2 radicals encounter each other and react to form a stable, non-radical product. For example, 2 peroxyl radicals might combine ROR, or 2 carbon-centered radicals might combine RR.
Are there ways to measure if this peroxidation is happening, specific markers?
Yes, several byproducts of lipid peroxidation can be measured as indicators of oxidative stress. Malondialdehyde, MDA, is a classic marker, often formed from fatty acids with three or more double bonds. Volatile hydrocarbons like ethane, primarily from omega-3 fatty acids, and pentane, primarily from omega-6 fatty acids, can also be measured in exhaled breath.
Thankfully, we have ways to control this. Antioxidants.
Absolutely. Our bodies have intricate antioxidant defense systems, and we also get antioxidants from our diet. These molecules help to control and reduce lipid peroxidation. In the food industry, synthetic antioxidants like propyl gallate, BHA, butylated hydroxyanisole, and BHT, butylated hydroxytoluene, are often added to prevent spoilage. Naturally occurring antioxidants are crucial in vivo. Vitamin E, alpha-tocopherol, is the major lipid-soluble antioxidant protecting membranes directly. Urate, uric acid, and vitamin C, ascorbic acid, are important water-soluble antioxidants. Beta-carotene can also act as an antioxidant, particularly at low oxygen pressures.
Are there different ways these antioxidants work?
Yes, they can be broadly classified based on their mechanism. 1. Preventive antioxidants. These reduce the rate of chain initiation. Examples include enzymes like catalase and glutathione peroxidase, which requires the mineral selenium, that break down hydrogen peroxide and lipid hydroperoxides before they can generate more radicals. Metal ion chelators, like EDTA or DTPA used experimentally, can also prevent initiation by binding metal ions like iron and copper, which can catalyze radical formation.
Yeah.
2. Chain-breaking antioxidants. These interfere with chain propagation. They react with peroxyl radicals faster than PUFAs do, donating a hydrogen atom to neutralize the radical and forming a less reactive antioxidant radical. Vitamin E is the prime example in the lipid phase of membranes, trapping RO radicals. Superoxide dismutase, SOD, works in the aqueous phase by scavenging superoxide radicals, O2FT. Urate and vitamin C also act as chain-breaking antioxidants in the aqueous phase.
Does peroxidation ever happen enzymatically?
Yes, while much of the damaging peroxidation is non-enzymatic autooxidation, the process can also be catalyzed in vivo by certain enzymes or compounds. Heme compounds, like those in hemoglobin or cytochromes, can promote it. Also, enzymes called lipoxygenases, found in platelets and leukocytes, specifically introduce oxygen into PUFAs as part of the synthesis pathways for leukotrienes and lipoxins, which we mentioned earlier.
Are there other significant oxidation products besides the peroxides and aldehydes?
Yes, two other important classes of oxidation products are often measured as markers of oxidative damage. Oxysterols are formed from the oxidation of cholesterol, and isoprostanes are prostaglandin-like compounds formed non-enzymatically from the peroxidation of PUFAs, particularly arachidonic acid. Measuring isoprostanes is considered a reliable way to assess oxidative stress in vivo.
Okay, one last major topic. How do lipids, being generally water insoluble, behave in an aqueous environment like our bodies? You mentioned amphipathic lipids.
That's the key. While bulk lipids like triacylglycerols are indeed very hydrophobic, water-fearing, due to their long non-polar hydrocarbon chains, many biologically important lipids are actually amphipathic. This means they have both a hydrophobic part, the nonpolar tail or tails, and a hydrophilic part, a polar or charged head group. Think of fatty acids with their carboxyl head, phospholipids and sphingolipids with their phosphate and other polar head groups, bile salts, steroid derivatives with polar groups, and even cholesterol with its single hydroxyl group, making it weakly amphipathic.
And this dual nature dictates how they arrange themselves when they encounter water.
Precisely. When amphipathic lipids are exposed to an aqueous environment or an oil-water interface, they spontaneously orient themselves to minimize the unfavorable interactions between their hydrophobic tails and water, while maximizing the favorable interactions between their hydrophilic heads and water. At an oil-water interface, they'll form a monolayer with the tails dissolving in the oil phase and the heads facing the water phase.
In a purely aqueous environment, they form specific structures, like bilayers in the cells.
Exactly. Depending on the shape of the lipid molecule and its concentration, they self-assemble into different structures. Lipid bilayers. Phospholipids and sphingolipids, which typically have two hydrophobic tails and a head group of roughly similar cross-sectional area, tend to form lipid bilayers. This is two layers of lipids arranged tail to tail, with the hydrophobic cores shielded from water and the hydrophilic heads facing the water on both sides. This bilayer is the fundamental structure of all biological membranes. Micelles. Lipids with a single fatty acid tail and a large polar head group, like fatty acids themselves above their critical micelle concentration or detergents or bile salts, tend to form spherical micelles. In a micelle, the hydrophobic tails cluster together in the core away from water, while the hydrophilic heads form the outer surface, interacting with water. Bile salt micelles are crucial for solubilizing fats during digestion. Mixed micelles containing bile salts and digested lipids help ferry fats to the intestinal wall for absorption. Liposomes. If you sonicate or agitate amphipathic lipids like phospholipids in water, they can form liposomes. These are spherical vesicles consisting of one or more lipid bilayers enclosing an aqueous compartment. They're essentially artificial membrane bubbles. Liposomes have potential clinical applications, for example, as carriers to deliver drugs or genes to specific cells. They can even be targeted by attaching antibodies to their surface, or in cosmetics and topical preparations. Emulsions. These are larger particles compared to micelles. An emulsion consists of macroscopic droplets of a nonpolar lipid, like triacylglycerol, dispersed in an aqueous medium. These are generally unstable and require an emulsifying agent, typically an amphipathic lipid like phosphatidylcholine or bile salts, to stabilize them. The emulsifier forms a layer around the oil droplet, with its nonpolar side interacting with the oil and its polar side facing the water, preventing the droplets from coalescing. Fat digestion in the intestine involves emulsification.
This has been an incredibly thorough deep dive. Just amazing how diverse lipids are. Let's try and quickly recap the key takeaways.
Sure. So the defining property of lipids is their relative insolubility in water, their hydrophobic nature. But many key lipids are amphipathic, allowing them to form structures like membranes. The major physiologically significant lipids include fatty acids and their esters, like triglycerols and phospholipids, cholesterol, and other steroids derived from it. Fatty acids come in different types: saturated, monounsaturated, polyunsaturated, and their chain length and degree of unsaturation critically influence the fluidity of lipids and membranes. Eicosanoids, derived from 20-carbon PUFAs, are potent local signaling molecules involved in inflammation and many other processes. Triacylglycerols are the main energy storage form. Glycerophospholipids and sphingolipids are the primary amphipathic components of membranes, but also have diverse signaling and recognition functions. Glycolipids are particularly important in nervous tissue and as components of the cell surface glycocalyx for cell recognition. Cholesterol is essential for membrane structure in animals and is the precursor to all other steroid hormones, bile acids, and vitamin D. And finally, lipid peroxidation is a source of damaging free radicals, implicated in disease and aging, but controlled by antioxidant systems.
That really drives home just how central lipids are to, well, everything in biology. Their unique properties enable so much structure and function.
Absolutely. Their ability to interact with both water and nonpolar environments and to self-assemble into complex structures is fundamental to life as we know it.
So thinking about everything we've covered, here's something to ponder. How do the diverse chemical properties we discuss, like the difference between a saturated fatty acid, a cis-unsaturated one, a phospholipid, and cholesterol, all come together within a single cell membrane to create a barrier that's both stable and incredibly dynamic and functional? How does that intricate mix allow membranes to do all the amazing things they do?
That's a great question to reflect on. It highlights the elegance of biological design, and of course there's so much more to explore, from the details of lipoprotein metabolism and transport to the intricacies of membrane-protein interactions and the specific roles various lipids play in health and disease states. Hopefully this deep dive has provided a solid foundation for anyone wanting to delve deeper into those fascinating areas.